This is a tutorial on how to submit high-throughput sequence data to GEO, the Gene Expression Omnibus. To get started, please go to our submission guidelines. A link to that is on the GEO homepage. This guidelines page includes detailed information on required files and submission procedures. Notice three required components of a GEO submission: the metadata, which includes the study and sample descriptions and protocols; the process data that were used to draw conclusions for the study; and the raw data. Note that raw data submitted to GEO is deposited to SRA for you by the NCBI staff. Metadata must be submitted in an Excel spreadsheet using our template. You can download the template by clicking on Metadata Spreadsheet, then on the button Download Metadata Spreadsheet. If you have already submitted high-throughput sequencing files to SRA and have created a bioproject, biosample, and SRA records for those submissions, please use the GEO Metadata Spreadsheet with SRA accessions to create GEO records that link the process data to your existing SRA records. However, most of you will be submitting raw and process data directly to GEO and should use the upper Download Metadata Spreadsheet button. We're now viewing that spreadsheet. On the Metadata tab, fill in the information for your study, samples, and protocols. You can access helpful tips about each field by hovering the cursor. It is important to provide information in all required fields, which are indicated with an asterisk. Incomplete metadata cannot pass the validation step on the GeoWeb upload page, and it will slow down processing of your submission. This template includes an Instructions tab that outlines all required steps and provides helpful links with even more information. And there are several example worksheets for different experiment types. We're now in a metadata template that contains example data. Let's review some tips for completing the template. In the Samples section, when you have paired-end FASTQ files, list both R1 and R2 files in the same row. And these paired-end FASTQ files must also be listed in this paired-end experiments section. Overall, check that your listed file names match the submitted file names, with all correct placement of dashes, underscores, and capitalization. Please avoid white space and special characters in file names. Use only alphanumerics, underscores, dashes, and dots. And remember that file names must be unique. Raw files for a high-throughput sequencing study are typically provided as FASTQ or BAM format. If you have a different raw data format, please check SRA's website to see if they accept your file type. Processed data should include the final processed quantified data used to draw conclusions for the study. For example, with RNA-seq studies, we are expecting the raw or normalized read counts for all analyzed genes. Please do not submit only a list of differentially expressed genes or read alignment files as processed data. After completing your metadata spreadsheet, get ready to transfer your raw and processed data files to GEO by FTP. Place all raw and processed data files in a single folder or directory on your computer for each experiment you are submitting. Choose a unique name for each folder, such as RNA-seq. Do not put FASTQ or BAM files in tar or zip archives. GEO staff must be able to directly access the files for processing. And do not upload subdirectories containing identically named files. You will transfer the folder to GEO by FTP. All of our instructions, credentials, and examples for FTP are available on this geo/info/submission FTP page, but only after you log into your MyNCBI account. This is what the Uploading Your Submissions section looks like after logging in. You will first create your personalized upload space by clicking the Transfer Files button. Then transfer the files to your personal upload space according to the instructions. After file transfer has completed, upload your metadata file. You can use this Upload Metadata button to get to the geo/submission/meta page. First, select the GeoFTP subfolder that contains the raw and processed data files listed in the metadata file. Then use Choose File to select your local metadata file. I've already done that. Next, select the date for your submission to be released for public access. The maximum allowable limit is 4 years from your submission upload date, but this date can be brought forward or pushed back at any time. Any GEO accession cited in a published manuscript or preprint, such as bioRxiv, will result in the release of the GEO records. Click the checkbox to confirm your release date. If you have additional information about your submission, you can add it to the Comment to GEO Staff box. After you click Submit, the metadata file will be assessed immediately for missing fields, sections, or other inaccuracies, and you will promptly receive a message with any identified errors and how to fix them. Once you have fixed the issues, please upload the revised metadata spreadsheet. Successful upload results in a message with a submission summary. You will also receive an email with this information, and your submission is placed into the GEO processing queue. Once processed, you will receive an email with the accession letter, including the GEO accession numbers and information on accessing your records and creating a reviewer access token. That's all for this video. If you have questions, please contact the GEO team at geo@ncbi.nlm.nih.gov.